So, next chapter is chapter 3. We, as a class, are a little behind the other classes. That's okay. Not a big deal. We will catch up. They took their exam on Wednesday. Some of them taking them today. We'll catch up. So, chapter 3 is all about cells. How many of you know cells already? Have taken cells in biology within the last 2 years? Okay, in high school. All right. So, a lot of this will be a review. We're going to get into a little bit more in depth than you may have gotten into in terms of in bio. And the first part of this chapter you already covered in lab, unless you have lab this afternoon, with microscopes. Okay? What we know about cells is based in what we call the cell theory. And these are your tenets of the cell theory. All organisms are made up of cells. All living things, right, are composed of cells. The cell is the smallest or the basic functional unit of life, or the smallest thing that can be considered to be quote-unquote alive. All right? All cells are related by their descent from earlier cells. All cells come from pre-existing cells. All right? So at some point you can go all the way back to somewhere. All right? And this last point is not really part of the cell theory, but lends itself to understanding how cells are related. Okay? Cells can be different. They have substantial differences in their structure and in their function. Okay? However, they do share common features. All cells have some type of genetic material. All cells contain ribosomes. Okay? All cells have a plasma membrane. Okay? So general features that they share, but they are quite different characteristics. Okay? So to study cells, we utilize microscopes. Right? Most cells are between 1 and 100 microns in diameter. Too small to be seen by the naked eye. How many of you had some cells for breakfast this morning? I did. 2, over easy. Right? So the smallest cell that you can see by the naked eye is probably a frog egg, which is about a millimeter in diameter. All right, so that's chicken eggs, ostrich eggs. Ostrich egg is the largest single cell thing in the world, blah, 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 blah. Unless you go back to dinosaur eggs, but we're not going there. Okay? So we need to have an assistance from technology, engineering, to actually be able to study and visualize cells. So that's why we use microscopes, microscopy, 2 types of microscopes. We have a light microscope, which you'll use in lab. Right, so I'm not going to go through the parts of the light microscope. You have that. You'll go through that in lab. You'll utilize it this weekend. Or this weekend? No, probably not. But this week in lab. And some of you have, what was I going to say? Some of you have your pond water that you're going to inspect, right, for little microorganisms. Right? How many of you have iPads, iPhones? Okay. If you go online, you can make your own. There's a tutorial to make your own microscope. Right, out of about $10 in parts if you really wanted to. Get a little laser pointer focusing thing, right? A plexiglass stage, blah, blah, blah. Professor Bartha actually has a model of one in her office that she made last year. So put the phone up there, take pictures of it. Cool. All right, so the point of the story is you can utilize the functions of light and bending light and lenses and physics to visualize very small things. All right, so with the light microscope, the light is transmitted through a specimen, it's refracted through lenses, and gives you a magnified image. All right, so important parameters of microscopy: magnification, resolution, and contrast. Okay, magnification is how much bigger are we making the image from what it normally is. All right, and how do we calculate magnification? Those of you who have had a lab or gone through it, how do we calculate total magnification? the objective number times what the typically 10, depending upon what the eyepiece is. All right, so the objective number multiplied by the magnification of the eyepiece. So objective magnification times eyepiece magnification gives you total magnification. Most of your eyepieces all the eyepieces you will use in lab are 10x. All right, so basically objective times 10 gives you your total magnification. Okay, what is resolution? Resolution is the measurement of the clarity of the image, right? Or the minimum distance between two distinguishable points. Resolution depends upon the quality of the objective lenses and the microscope itself. All right, so if you can resolve between two things, two different threads, right, you have a certain resolution. Right? Being able to tell the difference between where the nucleus ends and the endoplasmic reticulum begins is super high resolution. All right? Lower resolution would be showing where the nucleus is, the difference between where the nucleus is and where the plasma membrane is. All right? So you can tell the difference between resolution. And then contrast, visual differences in parts of the cell. So using contrast, you can actually see where the nucleus is because it's more dense than the cytosol, less dense, and so on. All right? So those three parts are important parameters of microscopy. All right, so here's what we can see based upon the type of microscope we're using or not using. All right, so height, human height, length of some nerve cells. You can see the length, but you can't see their width. All right, chicken egg, frog egg is right at the boundary of where we can see. Okay, and then the light microscope, most plant and animal cells, you can see bacteria. You can get to a point where you can see a mitochondrion, but that's pretty much at the limit of it. And then we utilize electron microscopy to see further smaller parts, cell parts. We can also utilize light microscope with certain staining procedures to visualize parts of the cell, and we'll go through that in a second. All right. Questions on why we use microscopy or microscopes? All right, good. So you can magnify effectively about 1,000 times. If you're using oil immersion, you will not be in class, so don't worry about it. And then you can use various techniques to enhance the contrast. All right, and then you see your organelles. Most of the organelles are too small, but you can sort of tell them out if you look close enough. All right, and then beyond that, organelles, we have to use electron microscopes. All right, electron microscopes function by streaming a beam of electrons across the cell, and because of when things are present, those electrons get absorbed. Okay, and as they get absorbed, they take up that path of electrons are disrupted. Changes in the disruption can give you resolution between things. Okay, so you have scanning and transmission electrons. So scanning electrons focus a beam. The scanning, or SEM, focuses a beam of electrons onto the surface of a specimen, providing images that look three-dimensional. So you want to see the surface of a fruit fly and the structure of its wings, you'll use a scanning electron microscope. Right? And you see the outside, or you want to see the structure of the plasma membrane. On the outside of the plasma membrane, SEM will show you the outside of the plasma membrane. Right? Or for the nucleus or whatever it might be. Right? Transmission electron microscopes focus the beam through a specimen. So it'll go through and you can see differences within. It gives you a two-dimensional picture. All right, because it's focusing the particle beam through the specimen, much like a light source goes through a specimen in a light microscope. Okay? So in the scanning electrons, you get a three-dimensional picture. In the transmission, you have a two-dimensional picture. All right, so what do they look like? Here's your different types of light microscopy. Bright field, whether or not it's stained. So bright field is shining light through the specimens, through the lenses, and you can make out a little bit of the nucleolus, a little bit of the nucleus, and the plasma membrane. That is based upon the refraction of light through differences in density of that cell. Okay? And then you stain it, you can see nucleus and cytoplasm differently. Phase contrast it's still the same light. Okay, but what they've done is they've disrupted how the light goes through the specimen. All right, so it doesn't go at a direct angle like you'll use in class. All right, just light comes through and directly shines through the specimen. Instead, the light sort of comes from either side or around it, and by doing that, you actually increase contrast, and you can see a lot different, some differences in the structure, better visualization of the nucleus and the plasma membrane. All right, and then you have differential and Nemarsky. Again, it's simple. It's along the lines of a phase contrast. More detailed, you get into fluorescence. All right, and this is what I like to do in my research protocols. I use fluorescent microscopy in class, or not in class, but in my research. So using different dyes, you can stain for the nucleus, you can stain for what you have done here, the ER, and parts of the cytoskeleton. You can look at those subcellular parts based upon different colors. All right, those different colors are a result of fluorescence, brightness, okay, that is in response to different wavelengths of light. Right? We know what light is a mixture of different colors, from red, orange, yellow, green, blue, indigo, violet, right? So by using certain wavelengths of that light, only red light, only yellow light, only blue light, you can then look at the fluorescence or the what we call emission of a certain dye. And it will fluoresce a different color. Confocal is along the same lines of fluorescence, but it increases resolution. So you see the difference between the left image and the right image. It takes several different pictures at different focal planes and then stacks them together to give you a super high resolution photo. And then you have your scanning and transmission electron microscopy. You see the 3D picture versus the 2D picture. Questions on microscopes up to there? Fluorescent microscopy, confocal. Okay. Recent advances. How else can we look at parts? Or say if I was interested in a particular protein that was changed in cancer cells. I want to look at retinoblastoma protein and what happens to it. What can I do? How do I find that particular protein? How can I track that particular protein? What do you think? A fluorescent marker to that particular protein. Okay? What do you know? And I gave you an example of this in a picture in the last, when we talk about the structure of proteins. What do you know that changes structure in response to what it's exposed to? Something in the human body that changes structure in response to what it's exposed to. Yes. But what in the human body? I think the picture was of a influenza virus protein. And I showed you structure was directly of this other protein was directly related to its function. And I showed you a ribbon structure of a influenza virus protein, and then on one side, and on the other side was a human protein. What was it? It was an antibody. Okay? So you are exposed, say it next time. You are exposed to proteins and antigens all the time. And those antibodies that you produce are changing in structure in response to that antigen. So if I exposed a rabbit to a foreign antigen, such as a retinoblastoma protein, that rabbit would produce an antibody which is specific to that antigen. Then I use that rabbit antibody, and I can follow where that protein goes. Okay? So by saying recent advances, we can label molecules and structure with fluorescent markers. So I take that rabbit antibody that's going to follow where that retinoblastoma protein is going to go. Now I put it on cells, and now I can just look and visually see where retinoblastoma is found in the cell. Pretty cool, right? I can see where actin is. I can see where actin is going because I have an antibody to it. Okay? And then you can use other stuff like confocal to increase resolution. And then you have more techniques for labeling. We have found there's a whole host of dyes that will permeate the cell membrane and collect in the mitochondria, or will collect in the endoplasmic reticulum, or the Golgi apparatus, so that we don't have to do these proteins anymore, the antibody stuff, or we just put it on and we can actually, in real time, as the cells are living, see what's going on. It's pretty interesting. You don't think so? That's fine. Other parts, other things that other techniques. Technique called cell fractionation. What do you think cell fractionation is, just by that? Breaking up cells. That's all it is. Okay? So it breaks up cells and you separate the components. A nucleus is more dense than the cytoplasm. Okay? Organelles within the cytoplasm are more dense than phospholipids. So if you break up the cells and then put them in this fluid and then expose them to centrifugation, spinning, right? Based upon their density, they will separate apart. So the nucleus is more dense, it will collect at the bottom. You'll have ribosomes are less dense, they'll be a little bit up, right? They'll all collect together. So then you separate them out. Then you can look at whether or not proteins are present in the ribosomes or where they are in the cell. All right? And this helps to allow scientists to determine functions of organelles. All right? And then with biochemistry and cytology, we can correlate function and structure. Questions on techniques and microscopy. All right? Don't expect to be examined on whether or not you know how to use cell fractionation. But if I gave you an example of an experiment, well, a research scientist did X, Y, Z, what is he looking at? You should be able to understand what I'm talking about and come up with, oh, he's looking at nuclei or whatever it might be. Okay? And for your quiz, I'll give you an example of that. Questions on that? So the next part is basically the next section in your book is determining what a eukaryotic cell versus a prokaryotic cell is. All right? There's a lot of words in there, and it's going to be a little bit confusing how they say it, but we're going to break it down. Okay? Basically, you have two types of cells, prokaryotic and eukaryotic. We know we have 3 domains within those two distinctions. Eukarya is one domain, which includes all eukaryotic cells, and then all prokaryotic cells are included in the archaea and the bacteria domains. Okay? Within that eukarya domain includes protists, single-cell organisms such as amoeba, paramecium, euglena, right? Fungi, mushrooms, yeast, okay? Animals, you and I, and plants, okay? They all consist of eukaryotic cells. Basic features of all cells, whether they be prokaryotic or eukaryotic. All cells contain some type of plasma membrane, a barrier between inside and outside, a separation between what is intracellular within the cell versus extracellular outside of the cell. All right? Within the cell, they have this semi-fluid substance called cytosol, which is, for the most part, a watery substance. Right? It changes depending upon which type of cell we're talking about and where that cell normally the normal habitat for that cell. Chromosomes. All cells have some type of, I don't like to say chromosomes because you think about some cells, they don't necessarily have multiple chromosomes or chromosomes in the way you think of them, but think of it as some type of genetic material. All right? DNA. They have DNA that is going to carry genes that will transmit or be the heritable information which will be transmitted from one generation to the next. Right? So some type of genetic material. And all cells contain ribosomes. What do ribosomes do? They make proteins. Okay? DNA is a great thing. It's a versatile thing. Okay? It includes it, but it's an instruction book. It's the user manual to your iPhone. Okay? How to use it. Ribosomes carry out let's change that. Ribosomes decode the code or read the instruction manual and produce functions so that you can text your buddies, Snapchat, put on Instagram, whatever it might be. Okay? So that ribosomes help produce the function. All right? So they make protein. Prokaryotic cells. Difference between the two. So all cells have those four characteristics, whether they prokaryotic or eukaryotic. Prokaryotic cells are characterized by not having a nucleus, or what we'll say is a membrane-bound nucleus. All right? Prokaryotic cells will have an area within them where the DNA or the genetic material is kept and clustered. It is not a nucleus because it is not membrane-bound. Okay? That area where the DNA is kept is called the nucleoid. There's no separation between the cytosol and the DNA at that point. All right? Bacteria have no membrane-bound organelles. All right? Most of the organelles in the eukaryotic, excuse me, the eukaryotic cells are membrane-derived, or they are surrounded by a membrane, except for ribosomes. All right? So you have mitochondria; they have a double membrane. The ER is a membrane structure. The Golgi apparatus, a membrane structure. Okay? And they have prokaryotic cells, cytoplasm bound by the plasma membrane. That is characteristic of all cells. Okay? Here's a diagram of E. coli. You see there's genetic material, a bacterial chromosome. Right? They have ribosomes. That clustered area, the more dense area on the electron micrograph to your right, the darker area is the nucleoid. That's the area where DNA is condensed. All right. In this case, bacteria have a plasma membrane that's internal to an external cell wall, and a capsule even beyond that. Right. And that's the structure of that is the subject for a microbiology class, not for this intro bio class. Okay? Point being, structure. Bacterial chromosome, ribosomes, plasma membrane. Differences, some cell wall, no membrane-bound organelles. Eukaryotic cells. The DNA is enclosed in a membrane-bound nucleus, and a nuclear envelope. All right? They have membrane-bound organelles, and the cytoplasm is your region between the nucleus and the plasma membrane. And in general, eukaryotic cells are much larger than prokaryotic cells, right? Because they have a lot more function. They have to do more things. Questions on prokaryotic versus eukaryotic cells? Okay. The plasma membrane, then, is your selective barrier that allows sufficient passage of oxygen, nutrients, and waste to service the volume of every cell. And how do those things move across the plasma membrane? Depends on what they are, right? How can they move past the plasma membrane? By which processes? I heard diffusion, yes. What else? What moves by, what is osmosis? The movement of water, right, in one direction or another. How else can we move things? Active transport, and the cell can also internalize or externalize things through endo and exocytosis. Right? We'll get to that transport in a little bit. The general structure of a biological membrane is a double layer of phospholipids, which means it looks like this. What is that? So double, these are phospholipids, right? Little diagram, here's your polar heads, your non-polar tails. So if it's a double layer of phospholipids, how am I going to draw, or someone take the iPad and draw the second layer? Who would like to do it? Nate, draw me the second layer of phospholipids. That's kind of fun watching. All right, so he's orientating the tails towards each other. Well done, sir. Why? Because the tails are hydrophobic and the heads are hydrophilic. So if this is the outside, and this is the inside, what is making up this inside out? What is making up the material that's mostly inside the cell or outside the cell? Mainly water, right? So water being charged as well. You have the hydrophilic portions arranged outside and the hydrophobic region internalized. Here's the basic structure. Transmission electron micrograph showing the double membrane. Okay? So the phosphate groups and the phosphate group on the head of the phospholipid as well as the carbons are more dense. Right? So you can actually see the double membrane, how the phospholipids are arranged. Okay? And you have your diagram. What else is enclosed within the phospholipid bilayer? Proteins. Okay? We have proteins that are wedged into the bilayer, and they have certain functions, important functions in terms of transport and cellular identification. So you have proteins, and then you have those. Some proteins have some carbohydrate side chains. Those are called glycoproteins. And beyond that, you have peptidoglycans or glycosaminoproteins, right? Or glycosaminoglycans, GAGs as it might be. That is a cell biology class. We talk about the structure of that. For this purpose of this class, there are proteins wedged within the phospholipid bilayer that have distinct functions. This is the structure of the plasma membrane, which is called what? Have you heard this term before? Whoop, that's a U. Fluid mosaic model. Okay, you've never heard. How many of you heard that before? Okay, the current structure of a plasma membrane, the model of the structure, the fact that the proteins are wedged in, is what we call a fluid mosaic model. Why is it a mosaic? What is a mosaic? Lots of pieces. What pieces do we have? Different parts do we have in the plasma membrane? Phospholipids and proteins, right? So together they function as a plasma membrane, so that in and of itself is a mosaic. We call it fluid because those proteins and those phospholipids are not stagnant. Okay? They're not stuck in one area. The proteins actually move across the plasma membrane. They float through it. Okay? So they can They're dynamic. They can change. All right? So the fluid mosaic model is a mosaic of phospholipid bilayer with proteins that is not stagnant, can move as necessary. Okay? Why are cells small? Why aren't all cells the size of ostrich eggs? More cells increase surface area overall. Okay? By increasing surface area, you allow the cell to do their jobs more efficiently. Okay? So the next couple slides go over what that means in terms of surface area. All right? By increasing surface area by a factor of 2, okay? So you go from 1 to 2, the volume within the cell increases by a factor of 3. Okay? The larger the cell, right, the volume is different. Okay? So it is the real importance of why cells are small is due to the need to keep the surface area to volume ratio at an ideal spot. All right? For those processes that we talked about for the plasma membrane to do, diffusion, transport, exocytosis, okay? For those things to work efficiently, the surface area to volume ratio has to be kept at a maximum. Look what it looks like. Okay, so here's the total surface area of one cell in the first column. All right, so if it's 1 unit high, 1 unit tall, one unit deep. Okay, total surface area for that square, we'll call this a cube, is 6. 6 faces of the dice, right? 1 by 1. Total surface area is 6. The volume is 1 whatever cube, right? Length times width times height. Yes? Okay. So therefore, based upon the surface area, the faces of the die, divided by the volume, your surface area to volume ratio is 6 for one of them. Now, I make that, now I have a fuzzy dice instead of a little die. It's a bigger cell. It's still one thing. Okay? Now, surface area increases. In this case, it's by 5. Right? So 5 times 5 times 5 is 125. That's your volume. Right? And 25 times 6, right? 25 being your surface area for one of the faces of the cube, multiplied by 6, 6 faces, gives you 150. Your surface area to volume ratio has decreased by almost a factor of 5, almost a factor of 6. So by having big cells, changes this surface area to volume ratio, which then alters how the cells function. They don't function well in transporting things, right? You have to consider having a nucleus wedged in the middle of this cell. Now, a protein has to get from the nucleus or from the outside of the cell to the nucleus. There are proteins that go from the cytoplasm to the nucleus all the time. Think about the distance that protein has to move. It's a lot larger than the distance it would have to move in the smaller cell. Okay? And now you break up this whole big cell into smaller cells, like a multicellular organism. You return the surface area to volume ratio back to 6. Okay? So maximizing the ratio between surface area to volume allows for efficient metabolic processes and functioning of the cells. And that's why cells are quote-unquote small. All right? Questions on this relationship? We'll stop there. Questions?